name's Andy Rathman Noonan. I am the Executive Director of the National Science and Technology Medals Foundation. We're incredibly pleased and humbled to be able to host this event, An Evening with Biotech's Best, here at USC in this beautiful facility, the Michaelson Center for Convergent Bioscience. Our foundation is dedicated to promoting scientific and technological excellence by connecting audiences across the country with recipients of the National Medal of Science and the National Medal of Technology and Innovation. These men and women are responsible for scientific and technological discoveries that have radically changed our world. Sometimes they impact in our lives in ways we may not even understand, we may not fully appreciate, but they're always there. So, with that in mind, I know it's a Wednesday night, it's hump day. You guys all have probably been working pretty hard this week, so we're gonna do a little bit of a warm-up, play a little bit of a game. So what I'm going to do is I'm going to ask you all a question. You're going to raise your hand if it applies to you, okay? How's that sound? Everyone good with that? All right, good. All right. So how many of you have gone grocery shopping? All right, so it looks like everyone has gone grocery shopping. Good sign. Okay, so Norman Woodland, he is a National Medal of Technology Innovation Laureate. He's responsible for the barcode technology you're going to find on your groceries. That helps track over $5 billion in product every year, including those groceries you're going to find at that awesome Trader Joe's you guys have at USC Village. I'm super jealous. There's only 2 in DC, so it's awesome. Anyways, let's stay on Trader Joe's for a sec, okay? How many of you love that freezer section at Trader Joe's? Okay, not as many as I thought. All right. Well, National Medal of Technology Innovation Laureate Frederick McKinley Jones is responsible for the refrigeration technology you're going to find in those trucks. They get that Mandarin orange chicken dinner you're going to find there in the freezer section. Make sure that doesn't spoil on the way over. How about, how many of you have washed your clothes in the last week? How about 2, so we get everyone's hand up? Okay, good. All right. Well, we have National Medal of Science Laureate Edith Flanagan. She's responsible for the filtration technology that took all the pretty harmful chemicals out of that detergent. That same technology is also applied to gasoline as well. All right, we're going to do a couple rapid-fire questions. Keep your hands raised as we move through. How many of you have a tablet, iPad, Kindle, what have you? Great. How about a fitness tracker? Awesome. How about a cell phone? All right. So everyone got their hands up at one point. All right. John Goodenough is a National Medal of Science Laureate. He's responsible for discovering the lithium-ion battery technology, which keeps all those electronics you've got powered, as well as thousands of others, including electric cars. I think it's pretty clear. From the discovery of life-saving cures to revolutionizing the field of telecommunications, the contributions of National Medal of Science and National Medal of Technology and Innovation laureates are far-reaching, extensive, and incredibly impactful. Tonight's event will feature 2 laureates and 2 more experts in the field of biotechnology. Our panelists are truly a remarkable group, and I encourage all of you to listen, please ask questions, and I hope, and for the sake of my foundation, I hope you're inspired. I really do. These are incredible stories, and we hope you get to hear them be told by these individuals. On a personal note, if you'll indulge me for a little bit, tonight's also a pretty profound occasion for me. 2 of the featured panel members, Dr. Leroy Hood and Dr. Marv Caruthers, worked with my grandpa, George Rathman, at Amgen. He's no longer with us, but he would have loved to be here tonight. I'd like to invite Dean Bradford to the podium for a few remarks. Thanks.
Thanks very much, Andy. You can recognize by the suit that I must be an administrator from the university, and I'm the person that's going to welcome you on behalf of the University of Southern California, and will really offer a warm welcome to the National Science and Technology Medals Foundation. Thank you for holding this event here and showcasing biotech best here tonight. And welcome to all of our visitors, both on the panel and the audience. It's a real pleasure, it's a particular pleasure for me to welcome you to this magnificent building. Michaelson Hall opened less than 4 months ago, and this building represents the joint initiative of 2 schools, the Dornsife College of Letters, Arts and Sciences, and the Viterbi School of Engineering. It opens up 180,000 square feet of new space, a hub for the instruments and expertise and cores of science and engineering on this campus, and a new way to do biomedical science. It brings scientists and engineers from what would normally be considered widely differing areas to solve really hard problems in human health, hence convergent bioscience. So this is one reason that USC is thrilled to join forces with the National Science and Technology Medal Foundation. We think this building and our Convergence initiative will help accelerate innovation in biotechnology and biomedical science and attract entrepreneurial faculty, postdocs, and students to our campus. Another reason we're really proud is we're very proud of USC faculty who've won National Medal Science and Technology and Innovation. So Andrew Viterbi and Sol Golom, both in 2007 and 2011, both winners of the National Medal of Science, and most recently Mark Humayun, who in 2013 won the National Medal of Technology and Innovation. So the four men and women you'll hear from tonight have each made remarkable impacts in the field of biotechnology. And I have the opportunity to introduce two of USC's own as participants in tonight's program. So first, Andrea Belz, who will be the moderator this evening. Andrea is the Vice Dean for Technology, Innovation, and Entrepreneurship at USC Viterbi. She serves as the Director of the NSF Innovation Corps, or I-Corps, which is funding an innovation node Los Angeles. And she served as entrepreneur in residence with faculty appointments in USC Viterbi, USC Irvine and Young Academy, and the USC Marshall School of Business. She also serves on the executive board of the Bridge Institute, which is one of the founding institutes in this building. Andrea holds a PhD in experimental nuclear physics from Caltech and a bachelor's in physics from the University of Maryland, College Park. And next I'd like to introduce Andrea Armani. She's a panelist tonight. Andrea is the Ray Irani Chair in Engineering and Material Science in the Viterbi School. And the mission of her research, a very dynamic research program, is to develop non-linear materials, integrated optical devices that can be used for portable disease, diagnostics, and telecommunication. She also directs the John O'Brien Nanofabrication Facility in the basement below us. And this is a major core that's being built up in this building. She received her bachelor's in physics from the University of Chicago and a PhD in applied physics from Caltech. She continued as a postdoc fellow there in biology and chemical engineering before taking up her appointment at USC. And finally, it's my distinct pleasure to welcome to the podium Amgen's Senior Vice President for Discovery Research, Raymond Deshays of Amgen. Prior to joining Amgen, and it's quite recent his move to Amgen, he was professor of Caltech and executive officer in Caltech's Division of Biology and Biomedical Engineering. He was an HMI investigator. In addition to his academic work, he co-founded Proteolix in 2003 and Cleve Biosciences in 2011. And as we do in these things, we give the academic pedigree. Ray's received his bachelor's degree in biochemistry from Cornell, his PhD in biochemistry from UC Berkeley, and he's a member of the National Academy of Sciences. Ray.
Judging by the lack of suit, you can tell that I work as an executive in a California biotechnology company. Thank you, Dean Bradford. It's my pleasure to be with you this evening, and I'd like to start out by thanking the National Science and Technology Medal Foundation for organizing this event and USC for hosting it in this lovely new building. I'd also like to thank all of you for showing up, and I'm going to conduct my own quiz. Can everybody who is under the age of 35 raise their hands? So it's a pleasure to see so many hands up because we have a lot of young people with us here tonight who hopefully will be stimulated by the words that they hear from our panelists, and they will go out and they will carry the torch that our panelists have initiated and will help find cures for many of the diseases that currently afflict us in society. Now, the two panelists that I will introduce, the first is Dr. Leroy Hood. He is an MD from Johns Hopkins Medical School and a PhD from Caltech, where he went on to become a professor. Now, at Caltech, Dr. Hood trained many, many luminary students over the years, and in addition made towering achievements in multiple fields, including in the field of immunology and gene recombination for generating antibodies, and also many, many achievements in the development of instrumentation. And one instrument that he developed led to the founding of Applied Biosystems jointly with Dr. Caruthers, who was the next panelist, and that was the peptide sequencer, which ended up being a very critical role, playing a very critical role in the history of Amgen, because it was the peptide synthesizers from Applied Biosystems that Lee and Marvin had founded that led to the sequence of peptides from erythropoietin that led to its cloning. And without that, Amgen essentially would not exist. Now, in addition to founding Applied Biosystems, that was in 1981, he had founded Amgen, again with Dr. Caruthers in 1980. And he's received many, many awards for his achievements. He's a member of all three academies, the National Academy of Science, of Engineering, and now the National Academy of Medicine. And I'm told there's only 15 living people in the United States that are members of all three academies. And in 2011, he received the national, 2011 or 2013, I think. 2013, he received the National Medal of Science from President Obama. And so it's really an honor to have him here tonight. You know, there is a story when I was an assistant professor, and you can now confirm whether this is fact or fiction, that in the early days of making, because one accomplishment I forgot to mention was the 4-color sequencer, which was the basis for the Human Genome Project. And I was told that you were experimenting with very long gels, and they were so long that they had to be done in the stairwells of the building of Braun, which is where I had my first laboratory. You're shaking your head, yes? That's very excellent. The second of our final panelists that I'll introduce is Dr. Marvin Caruthers, and he's distinguished professor of biochemistry at University of Colorado in Boulder. And he did his PhD at Northwestern in Chicago and then went to do a postdoc at MIT with H. Gobin Khurana. As a chemist, he was the person who really launched the modern age of gene cloning by developing the methods to synthesize oligonucleotides using phosphoramidite chemistry that his laboratory developed. And it's hard to appreciate how important this really is nowadays, because these things are all taken for granted. But the ability to synthesize probes like that allowed information taken from peptide sequencers of the sort that Dr. Hood developed, and then enabled protein chemistry to be translated into cloning of genes through synthesis of oligos from those peptide sequences. And I gave a seminar in Boulder last week, and I had a meeting with Dr. Thomas Cech, who's director of the Biofrontiers Institute. And there were two notable things about that visit with Dr. Cech. The first thing is walking in the building, because the building is named the Jenny Caruthers building. And so a very generous gift made by Dr. Caruthers in honor of his wife who had passed away led to the construction, or underlay the construction of that building, which I was told is the second biggest building on the entire campus, second only to the football stadium. The second was that Dr. Cech told me that Dr. Caruthers supplied the oligos that led to the cloning of erythropoietin. And Dr. Cech told me 30 oligos, but I was talking to Dr. Caruthers earlier tonight. He said it was probably more like 400 or 500 oligos. And again, you know, without that contribution, Amgen and all the patients that Amgen has served over the years simply would not exist. And so, it's really with this sense of humility that I welcome all of our panelists to the stage to begin this discussion. Thank you.
Hi. It is such a pleasure to be here tonight. As you heard, my background, I want to be very fair in terms of being one of the biotech's best. I took a year of general chemistry with the pre-med majors before running to the physics department. So I want to be very fair about that, although I've had the opportunity to work in multidisciplinary groups since then. And this is intended to be an opportunity for you to hear some of the real stories, not just the extensive bios that you can read online. There's so much information about all of our distinguished speakers here, but for us to learn a little bit about the real path that each one of these people walked alone or together. But I'd like to start with just an opportunity for each of our speakers to give you a brief synopsis of their extensive accomplishments. Do you want to start?
Sure. So what I'm really passionate about is using systems-driven 21st century medicine to revolutionize healthcare. And that's going to do three things. One, it's going to improve the health of the individual. Two, it's going to make medicine truly predictive and preventive. And three, it's going to enormously decrease the cost of healthcare. And I'll tell you in a moment what 21st century medicine is about. When I started at Caltech, I was really impressed with the complexity of human beings, and I'd always wanted to study human biology. And that really drove me to thinking about how you dealt with complexity and led to a discipline called systems biology. And in fact, in 2000, I started the first Institute for Systems Biology. And it's about a holistic approach to analyzing systems. It's about global accumulation of data. It's about the ability to integrate the data to mimic the biological networks, hierarchical in nature, that drive development and physiology and aging. And most important of all is about this simple idea that biology is an informational science. And in the early 2000s, we focused these systems thinking on disease and came to three really fascinating concepts about healthcare. So the first, applying systems thinking to disease, led to a discipline we now call systems medicine. And driving essences of systems medicine are, one, the ability to develop relevant new technologies that open up new dimensions of human data space so we can explore the unknown. And number two, developing the computational tools so you could deal with the data and lead to sensible conclusions about what the experiments really meant. The second realization was that healthcare should be predictive, preventive, personalized, and participatory. And we call this P4 healthcare. And the essence of what P4 healthcare says is really revolutionary for modern healthcare. It says there should be two major thrusts in healthcare. One is wellness, and one is disease. And wellness is almost entirely ignored today. But for compelling reasons in a 10 to 15 year period, wellness will be the dominant thrust in healthcare because, interestingly enough, it is the key to understanding disease. And I'll explain why that's true in just a minute. Now, the third major concept in thinking about wellness was the realization that humans are incredibly complicated organisms. And when you go to the physician, you're lucky if you get 15 to 20 observations, clinical tests, and that doesn't even begin to approach the dimensionality. So we set up an experiment that advanced into a company where we created personal, dense, dynamic or longitudinal data clouds for each individual over the period of a year, where the data clouds numbered in dimension in the billions of data points. And we demonstrated over the course of that year that we could integrate these data together, which led to actionable possibilities for improving wellness and or avoiding disease, and started a company called Aerofail that now has 4,000 individuals that are doing the same thing. But the really important point was, as we accumulated the longitudinal data, we began to see state transitions from wellness to disease for all chronic diseases. We can use the data clouds to obtain the biomarkers that mark the earliest transition, and then systems approaches to generate the therapies that will reverse it. And the first disease we're attacking with this approach is Alzheimer's, and I'll be bold enough to say in a 3 to 5 year period, we'll be able to reverse 80% of early Alzheimer's back to normal. And we think we can do that for all chronic diseases. So this is the predictive preventive disease of the future. So what is 21st century medicine? It's the combination of these three things: systems medicine, P4 healthcare, and scientific wellness that focus together to transform how we practice healthcare today. And again, you can begin to understand why I think the 21st century medicine will, one, fundamentally change the quality of health for the individual. Two, will bring in prediction and prevention, and in time, the end of most chronic diseases. And three, it's going to really, getting rid of chronic diseases alone will enormously decrease the cost of healthcare.
Well, there's certainly a compelling vision, is an understatement. So we'll have a chance to delve deeply into this. Andrea, I love the fact that Lee started with the statement of what he's passionate about. So I know that you have such an exciting laboratory and so many interesting thrusts to your research. Tell us about that.
So I'll try to keep it to 2 minutes. Be super fast. So I'd say there's kind of two main passions I have. The first is, as a professor, I'm obviously passionate about my students. As running a research group that are filled with students, that's one of my main loves, is trying to train the next generation of me's, the next generation of researchers and leaders and faculty or industry members. And that's one of my main passions, whether they're K through 12 or undergrads or grad students. As well as my particular research area, it actually aligns very much with what you mentioned, but focusing mainly on, instead of trying to create ever-increasing, complex, and expensive instrumentation for doing diagnostics, kind of trying to go the other way, right? Maintain the level and the performance we have now, but trying to leverage the advances in computational and signal analysis while decreasing the complexity of the instrumentation, making it smaller, making it lighter, so that we can have more instruments, so we can have them in low resource areas, so we can have them in rural areas, so they're easier to use and easier to understand the results. So trying to address both problems at the same time, because we don't necessarily need something that's larger to actually make it better.
And that there's a marketplace for both of those, right?
Exactly, there's a marketplace for both.
There is one market for the most sophisticated instrumentation, and then quick and dirty. The reason people talk about Band-Aid solutions is that Band-Aids work.
Band-Aids work.
So that's really interesting. Marv, tell us what you really care about.
Well, I'll start with follow-up, before I start on that, a follow-up on Andy's comment about different Medal of Science winners and what they developed and things like that. And it was mentioned earlier that we developed the chemistry that everybody uses for synthesizing DNA. That's in most of the biological laboratories, biochemical, molecular biology laboratories in the world. Whether that's in biotech, pharmacy, government labs, etc. A good fraction of the basic research in the biological sciences requires synthetic DNA. But it's been around, we've had it developed now for quite a few years, and sometimes, if it's the right audience, I'll introduce the topic. I don't even talk about synthetic DNA much anymore. But I'll say, well, you know, most of you as young faculty researchers probably think that FedEx developed the chemistry for synthesizing DNA. Because what you do is you go to your computer and you type in a sequence and you hit the return button, and the next day FedEx delivers your DNA to you. So FedEx must have developed that chemistry. What am I interested in these days? Well, it's still a lot to do with the area of nucleic acids. For example, I've been working the last 4 or 5 years with Agilent, where we've adapted our chemistry to instruments where we deposit DNA on chips. And we can make oligonucleotides now anywhere up to 300 in length. And we make every day on these chips 250,000 pieces of synthetic DNA. And every day they make billions of pieces of synthetic DNA in that size range for various applications all over the world. One of the applications that really intrigues me, and this has nothing to do with biology, is storage of information. For example, and there's been a few studies so far in this area, where calculations have been made that you could store all the information that's ever been accumulated in the Hubble telescope field on 50 milligrams of synthetic DNA. And you could deposit all the petabytes of information coming out of the CERN laboratory on 35 grams of synthetic DNA. Now, our chemistry isn't there yet. We can make 300 mers, but we have to figure out how to make 1,000 mers, and that's not an easy task. And there are a lot of challenges there, and that's one area that interests me, is to see if we can at least develop the chemistry basis for storing information on synthetic DNA. So that's of interest to me. Another area that's really been a lot of fun lately is we figured out some derivatives of DNA that have a redox potential. And we can therefore deposit in a defined way on duplexes of DNA, gold, silver, platinum, palladium, metals like that. And we can imagine maybe in the future, I'm trying to tell you where we're going. I'm not showing you any of these where we are at. I'd like to figure out if we could make wires out of synthetic DNA where we could connect the strands with metals between 2 electrodes, and we'll see how that goes. Because we now can clearly make synthetic DNA derivatives that will deposit very clearly, right in the spot where the synthetic DNA is located, a metal, a conducting metal, like silver, gold, or platinum. So those are 2 areas where we're going. We keep playing with nucleic acid analogs, because there are quite a few potential applications that require synthetic DNA in a therapeutic sense that's out there. And recently, we've started into a group of analogs where we can actually do a lot of research on exon skipping, which is responsible for a lot of genetic disorders. And currently, we have one analog that's far, far better than anything else out there for recovering the correct gene sequence in Duchenne's muscular dystrophy, which is a classic example of how to proceed with using synthetic DNA for these types of applications. And one area where you mentioned a simple diagnostic for the third world, and we think that we're trying to develop these DNA probes that reduce metals so that maybe we can apply them in a simple sense to a diagnostic that can be used in the third world for diagnosing all kinds of viruses and simply reading it off of a cell phone detector, you know, when you look at the picture and you can see what's going on. So we're developing those kinds of areas at the current time in the lab. So I think I'll stop at that point.
I want to talk a little bit about the path by which you got here. And I think one thing that I have seen and that I remember from when I was young was being a tinkerer. I had a Radio Shack circuit kit that made lights blink and you could make a bell ring. And so I wanted to explore your tinkering background and how that informed your ability to invent later. And so actually we'll start with Andrea. Tell us about your tinkerer background.
So one of the first toys I actually remember as a child were Lincoln Logs. So everyone else asked you, how many of you had Lincoln Logs? Yes, I was not the only one.
More people than said yes to grocery shopping.
Yeah, exactly. Yeah. Excellent. So they're basically, for those who don't know, like large toothpicks that you can stack into arbitrary shapes. The goal is to make cabins, but I also made teepees for whatever reason. And I think just the idea of being given a bunch of straight sticks and having the freedom to create whatever I wanted to was, and build whatever I wanted to I was 2 or 3, kind of set my mind into that construction and building mindset. And then the toys I got after that were things like Legos. So I essentially inherited my uncle's and my father's and my grandfather's toys, as opposed to buying other toys. And so just having a very hands-on constructionist toy set really, I think, influenced my later career.
Lee, did you have any experiences like that as a kid? What did you build?
Well, my experiences were more theoretical that had a profound impact on me. So my father was an electrical engineer that worked for AT&T. I grew up in Montana. And every summer in my high school years, he would go to Boulder and teach courses in electrical engineering to men throughout the company. And he always had me come and take these courses. And so I learned kind of circuit design and systems thinking. And those courses, to the extent I understood them at the time anyway, really profoundly changed how I thought about biology. And that's why I got interest. Over the years, we've developed 6 different instruments that read and write DNA in various ways and various forms. And increasingly with sophistications that increase throughput, miniaturize the use of reagents, make things less expensive, and so forth. And I think a lot of that came from that engineering view of life that I superimposed on biology.
That's really interesting. So you're informed by this early exposure to how circuits and dynamic systems operate, and that would inform how you think about feedback and other dynamics like that. And Andrea's informed by putting things together and building something out of simple elements. Marv, what about you?
Well, I was always interested in science, as far back as I can remember. And I can remember in the 3rd grade, my parents bought me a chemistry set. And of course, I did all the experiments and things exploded and all that kind of thing. And after that, I thought, gee, I know everything there is to know about chemistry. So I think I'll go on from there, But then my best friend and I had a chemistry magic show in the 6th grade, where again, we had things exploding and colors change, liquids changing colors and all that kind of thing. And in those days, I'd read every science fiction book in the Des Moines Public Library. I grew up in Des Moines, Iowa. And waiting for the next issue of the Scientific American to show up in my mailbox, because that's the kind of life I wanted to live. And I was really lucky in high school because I had a science teacher. His name was Paul Sloan, who organized a science club for us. And he took us around to, different hospitals and different analytical laboratories and things like this all over the city to stimulate our interest in science. And I grew up in a very small class. This was a very small high school in the suburban Iowa, Des Moines, Iowa. It wasn't one of the bigger schools inside the city. We only had a class of 28 graduates. But out of that class, we had one who became a nuclear physicist, graduated with a PhD from Berkeley. Another got his MD, PhD from Hopkins. And one became an aeronautical engineer out here at Langley in the aerospace division. And it goes on and on. Most of the kids in that class were really pushed the limits in terms of what we could do. And when I got to college, I found out that, my goodness, these kids aren't as well trained as we are, and we came out of high school, you know, because the competition in that class was very high. So I've always been interested in science, and that's the direction I've continued to go in. I always thought I'd end up in either the biological or physical sciences, and that's where I ended up.
And it sounds like it was driven in part by the people with whom you were surrounded. So let's fast forward a little bit as you began building your university careers. And tell us about, Marv, maybe we'll start with you since you touched on this, the search for great collaborators. And you can remind us maybe how many years you and Lee have worked together. I can take a guess, but I'd love to hear the real answer. But tell us a little bit about finding people to work with who can help turn you into a better inventor. How does that happen?
Well, you know, when I joined the faculty at University of Colorado chemistry department, we had a faculty. The year before, there was a biochemistry division, I think 7 or 8 out of 25 or 30. And 3 of them left, 1 to the molecular biology department and 2 at SUNY, New York. And so there were 4, and they were all senior people who were pretty much close to retirement and weren't really very research-oriented. And they brought in 3 of us at the time in biochemistry, and I took the lead, as is usually the case in those days. And we started building that department and division to the point where it is today. And we hired some and it was mainly, I pushed it really hard, and I got a career development award, and we all used that money to bring in a lot of first-name speakers around the country. And then 3 or 4 years later, we hired Tom and Carol Check, and we hired Uhlenbeck, and pretty soon we had a really top-rated department. I didn't start, when I started there, we weren't working on nucleic acid chemistry at the time. As a matter of fact, the first 25 or 30 papers we published were in the area of how to understand how proteins recognize DNA sequences. And it was only 4 or 5 years after I started there that a graduate student came into the lab, Mark Matucci, who wanted to do a PhD in organic chemistry. So I thought nobody really developed the chemistry yet for synthesizing DNA that everybody could use, so let's give it another shot. And so we started working on this, and a couple years later a postdoc joined, that was Serge Boucage, and we developed the chemistry that everybody uses. And sometime around 1980, perhaps, 81 or 79, because Bill Wood was a good friend of yours who had moved to MCD biology. Lee was on campus, and we met, and I showed him what we were doing, and that was our first encounter. And the second one was actually when Winston got us involved in starting Amgen. So I probably, the date from which you and I kind of started to get to know one another was probably either 79 or 80 or 81, sometime in that period of time.
Oh. And in your second meeting, you decided to start Amgen. That's a pretty
it's just not the two. I'd say there were 7 or 8 scientists, and 3 venture capitalists can can lay claim to being co-founders of Amgen. I think
Absolutely, yeah.
That's what you would
it
say.
it was a group crowd.
It was a group.
In fact, the guy that was the real leader initially ended up backing out, and all the rest of us carried through and started
right.
yeah.
That's interesting.
Yeah.
And so maybe you can talk a little bit about the process of working intensively with a group. You said it was 7 or 8 scientists and a couple of investors, but talk about those early days and working in a collaboration like that outside the university. What was that like?
You know what I'd really like to talk about is the early days of technology development, but I can do that one too, okay?
We have...
I have permission.
Well, I was going to say we have plenty of time. Permission, I think, also, yeah.
Well, you know, what was really interesting is when I went to Caltech in 1970, I told my chairman, look, I really wanted to do 2 things. I wanted to do technology development because, I'd been thinking about complexity and data in humans and realized we really needed machines for doing this kind of thing, and molecular immunology. And what was really, and my idea was I'd really recruit terrific grad students to become postdocs from organic chemistry, from engineering, from computer science, and I was really successful in doing that. And I ended up getting people like Mike Hunkapiller, who really pioneered protein sequencing, one of the really revolutionary tools for interesting reasons. And then Lloyd Smith, who really was one of the leaders in doing automated DNA sequencing. But what was really interesting about Caltech was at year 3 in '73, I had the chairman come into my office and say, I'm here to urge you in the strongest possible terms to give up all of this technology. And I said, I wasn't going to do it. It's really going well. And 25 years later, I asked Sinsheimer why he said that. And he said it because all the senior biologists thought it was unseemly to have engineering in biology. And he said, what they really urged me to do is transfer you to engineering, which I never would have agreed to, and Bob didn't ask me to do that. But this whole idea of really good departments, and biology at Caltech was really superb. But you know, they become arrogant and they become narrow-minded about what's really good science. And I think that was a really interesting lesson. And I'll tell you, I saw it again when I started pushing in '85 the Human Genome Project. Again, the senior people were fanatically against it. And the essence of the argument was, big science can never be good science. It's going to take money away from what we do, which is little science. And hence, and people were unable to hear the utterly compelling arguments that existed. And I realized it's always true. We filter and hear what we want to hear, no matter what the argument is. And you know, one of the paradoxes I've really struggled with in my academic life is how do you convince people, if you're ahead of the curve, that what you're doing is legitimate and meaningful and important? And frankly, that is exactly the reason I left Caltech. I felt in the end that the 2 things I valued the most, the department had said this was really inappropriate. And, you know, frankly, Caltech missed the genome revolution completely, which is an enormous shame because they could have had a lot to contribute. But what I was able to do is, with younger people, really build up an absolutely superb engineering, computer science, cross-disciplinary group that made it possible to give an individual a responsibility for taking on an incredibly important instrument. But he had, or she had, all the help to put these things together. So it was, in my case, for all the technology development, they came out of my students and my postdocs.
Well, you're touching on different elements of collaborating and being inside of a research organization. and how important it is to really surround yourself tightly with the right people. I mean, you're describing a group that certainly does not sound like your typical biology group, right? If it's got engineers and computer scientists in it. I think, Andrea, you've also built a group that does not look like any other on campus. And maybe you can talk about your ideas of how you really foster a cross-disciplinary approach that doesn't sacrifice any rigor in the service of crossing the disciplinary lines.
So first, we're at USC. That really helps. And we're also located in the Michaelson Center, which one of the goals of this building is actually to do exactly what you described. So it's to try to bring together all of the disciplines and have all of the researchers talk with each other. So while my group does have researchers from electrical engineering and material science and biology and chemistry and biomedical engineering, and I'm going to forget a discipline and then that student's going to feel left out. I'm really sorry. In the lab right next to me, there's a computer science group that's doing bioinformatics, and the lab next to them, there's an electrical engineering group who's working on implantable devices. But when I say lab, what immediately comes to mind is walls, right? Upstairs, literally 3 floors up, are my labs. There aren't walls. It's more describing in this open floor plan space where my students are, that's where they're doing research. And then in this kind of nebulous, fuzzy area, you move over, and then there's the other researchers. So yes, my group is in and of itself interdisciplinary, but then they kind of blend over and merge with the other students who are also interdisciplinary. So then you just end up with this gigantic melting pot of researchers doing all similar but yet different things, and they all learn from each other. And if I have a biologist who is trying to do some programming and she's struggling with it a little bit, she can go over and find a computer scientist who can teach her how to do the programming. Similarly, the one frustrating thing I've had with a couple of my electrical engineering students is they've forgotten biology. Most importantly, no.
Or never knew it.
Or never knew it. For example, cells die. Yeah. This seems to be a concept that was never taught. But I can just, when I have one of them come into my office, I can be like, no, I'm not going to teach you biology again. Why don't you go talk to Sam? And they will just go, they'll get a crash course in biology, and it's very easy because they have all of each other to learn from. And that's like one of the beauties of USC, but also this building. So that's why I'm so happy in my new home.
And Marv, you described a couple of different applications of your research that are really outside what one might think of as traditional chemistry, right? You have these effectively these electrodes, these DNA electrodes that you're discussing, as well as DNA as which we know it's a storage medium already, but really harvesting that much more deliberately. How do you build an interdisciplinary group that can attack those problems without running into some of the organizational challenges that Lee very candidly described? Thank you. So how do you forge that kind of mix in your group?
Well, I also have had a lot of the organizational challenges that Lee mentioned as well.
You're among friends.
You're among
Tell us.
But currently in the department or the building, this new building we have, it's designed along lines that you mentioned. We have chem engineers, and we have the biochemistry division, which will soon be a separate department. And then we have Tom Check's BioFrontiers Institute. And the BioFrontiers Institute has within its framework theoretical biologists, it has mathematicians, physicists, chemists, biologists. So within this same building, for example, right across the hallway from my lab is the chemical engineering group. We have people integrated much like you described. So that helps a lot. And, my group has always been quite diverse. I've had chemists who became biologists, and I remember one biologist who joined my lab, and he said, I don't want to do any biology, I want to learn some chemistry. So I put him in the chemistry lab and said, go to it. And that's what, so I've integrated these different functions And I always try, especially with PhD students, to give them a project where by the end of their stay, they'll have done some biology as well as some chemistry. And that's just the modus operandi. And every student has his or her own project, so it operates like that. But coming back to a couple things that Lee said that I think I want to elaborate on in the nucleic acid field is that, you know, it's not as though the whole world of biology and biochemistry, et cetera, were waiting around on the edge of their chairs waiting for us to develop a method for synthesizing DNA. That was not the case at all. As a matter of fact, most people in the biological sciences couldn't care less. I remember when I was, in 1975, I attended my first Gordon conference. And by then I had a reputation in this field, and the organizer asked me to give a review of nucleic acid chemistry, so I did it. And then that night, afterwards, we were having a beer around a table, and a person who's now a member of the US National Academy, and I won't mention his name, was sitting there, and he said, Marv, he said, why the hell do you want to learn how to synthesize DNA? He said, yeah, Khurana used it to solve the genetic code, and now he's made a gene, but what else are you going to do with it? Why don't you go off and try to learn to do something more interesting? And that was not unusual. That's the way it was in those days. Nobody cared less about, they couldn't care less about these kinds of approaches. And as you mentioned with what you've been doing, I mean, you know, you're out there doing what you know is important, despite the fact that the general science community really thinks what you're doing is kind of dumb and not very important at all. Every once in a while, I drag out a slide of mine that I only use it these days if I'm in a meeting in England where I know there's an editor of Nature or a Nature correspondent who's in the audience. And this little slide is, I somehow work it into my talk. It's a slide it's a quote from an editorial in Nature, which was published shortly after we had synthesized the first gene in Khurana's laboratory. And this correspondent, unknown who it was, wrote at the end of this little synopsis where he was being rather caustic about the whole approach, said, and now Khurana, like NASA with their Apollo program, has synthesized the gene and the feat will never be repeated. You know. So I love to, sometimes I'll bring that quote up if I know there's a Nature correspondent in the audience. But it's you know, it's this kind of an attitude that if you're trying your best to develop new methodologies, Lee and what he's doing now, and us trying to develop methods for storing DNA on chips for information, storage of information using synthetic DNA, et cetera, et cetera. You know, people look at that and say, well, you know, there's all kinds of disk out there now for storage information. Why do you need to develop DNA as a storage device? And I've got my reasons, and I think they're good ones, but most people just kind of shake their head and say, well, that's kind of ridiculous. Why are you doing it? But I think you just have to be a little bit stubborn sometimes and think you know what you're doing. And then eventually, some of your ideas actually work. And not all of them, of course. That's for sure.
Can I add one thing to that, too? Because, you know, I had exactly the same kind of experience with the DNA sequencer. I remember in about 82 or 3, Jim Watson and I were talking, and Jim said to me, Lee, why are you wasting all of society's money on this DNA sequencer? He said, number one, graduate students can sequence DNA really well, and they'll be able to sequence all the DNA we'll ever need. And he said, number two, think about it. If we use the Chinese population that was then 1 billion, each individual would only have to sequence 3 nucleotides, and we'd be done with the Human Genome Project. I sent my first 2 grants on the sequencer into NIH and got the worst priority scores I ever got, with comments like, this is impossible, and on and on and on. But I think the key thing for the students is I actually went to a foundation and went to a company and persuaded each to put $200K in. We had enough money to develop the prototype and prove it worked. Then you could, once you've proven something to NIH, they'll fund it. They just don't want to do the discovery part of stuff.
Absolutely.
So,
that's absolutely true.
yeah.
A well-known scientist, and I definitely will not tell you who it is, when Sanger was learning the first methods for sequencing DNA, the plus-minus approach, this guy said, why the hell is he doing this? He's going to get a bunch of A's and G's and C's and T's, and what is he going to do with them?
Yeah. Anyway, we've made the point.
We've made the point.
Yeah.
I want to go back to a word that you were using earlier, which is the word systems and systems thinking. And physicists, of course, love this because we're always trying to create more grand unified theories. So the more we can integrate into one simple equation, the better. But let's talk a little bit about what that really means, especially because we have so many students here who maybe are not used to looking at something from a systems perspective. It can be challenging sometimes when you're working on your narrow problem that you need to solve entirely. And so, Lee, I am going to start with you on this. I suspect you have something to say about systems thinking. And tell us, how do you teach systems thinking when it's very difficult to generate a global perspective as a student working on a narrow problem?
Let me interject. What you do is you invite Lee to come and talk about it for about 2 or 3 lectures, and he'll have everybody convinced.
He'll have everyone signed up.
Yeah.
We may get some of that text of those 2 or 3 lectures here. We'll see.
Okay, the easiest way to explain systems thinking is to think about the famous Rube Goldberg cartoon that attached together 14 different devices in a very complicated manner for Rube to be able to cool his soup. So that's a complex system. So what would you do to understand how that system worked? Well, 1, you have to define the components in the system. 2, you'd like to know what the components did individually. 3, you'd like to know how those components were connected. You'd like to know the dynamics of the motion that led to the soup cooling. And you'd like to understand how the process is regulated. So that's a simple analogy of what we have to be able to do. And that's why I said, when you go to the doctor's office and he makes 20 observation measurements, I mean, he's so far short of being able to assess 98% of the things most of us would really like to know about. So it's this idea of taking a holistic view of the system, of doing the global comprehensive analysis. And of course, in living systems, the information is driven by biological networks that are hierarchical in nature, and you have to be able to integrate the data so you can recreate those 3-dimensional networks and their operating and things like that. So in a simple sense, that's how I think about systems. But it's a really 5 or 6 part simple algorithm. What are the components? What do they do individually? How are they connected? What's the dynamics? What's the nature of the regulation?
And it does sound clear how you could draw the line from the early electrical engineering lectures.
Absolutely.
Absolutely. Those are the same kinds of systems and the same questions you ask is what does the feedback and what do the dynamics look like? So how do you teach that effectively, Andrea? How do you teach people how to really look back and understand all of these regulatory processes and time scales and all the other questions we ask?
Very challenging. So I think it also depends on the student and the student major, right? For example, if it's a double E, it's really easy. You can use circuit theory. If you are looking more at a biologist, unfortunately, it's not in their standard course curriculum. So then you actually have to actively teach it as part of research. And you do have to, invite Lee to give a lecture in your class. Apparently, you're going to get a lot of speaking engagements. But you could also, start with a simple experiment, right? Like one experiment, and then start looking at how the experiment changes with time. And really, in biological systems, time is that critical factor that physicians even seem to overlook. If they take blood in the morning versus in the evening, they could get a different result. And if they don't actually monitor that they're taking your sample sometimes at 9 a.m. and sometimes at 4 p.m., that's an important variable.
Yeah.
So, and it goes back to the tinkering of if you want to understand the system, you may as well build it and touch it or model it, which is really what the circuit mapping tells you. So how do you teach in chemistry and the chemistry of biological systems? Is it easier to teach systems thinking because you're used to looking at systems in equilibrium and so on? Or do you find that you still are augmenting the traditional chemistry curriculum with the way you look at the world?
Well, historically, chemistry has been inorganic chemistry, organic chemistry, biochemistry, well, recently biochemistry, analytical, theoretical, okay? And the twain shall never meet, and they kind of speak to one another in the hallway, but they never interact. But now, it's, even at the university level, it's a system where you've got not necessarily chemists talking to other chemists in one field or another, but you've got the university community working together where biologists try to understand chemistry and vice versa, or you throw in a physicist who's interested in biology, and pretty soon you're trying to speak the same language, which in science, as we all know, is difficult because even subdisciplines have a jargon that's so different and difficult for one group to learn from the other. And I think these barriers have been broken down over the last 15, 20 years maximum, where science is a community effort on most campuses these days. It's not a departmental story or a sub-department level. And it's almost like a university experiment in systems science one thing I've always admired about Lee and the systems biology approach is that to put together a group, a large group, an institute, where you're looking at very important factors, but in order to understand, not factors, but different important aspects of biology, but in order to do that, you have to bring together a group of scientists from all kinds of disciplines, and they have to learn to talk to one another. And they have to understand that this is the objective. And there aren't too many places and leaders in the world who have the ability to organize like that, and Lee is one of them.
It's interesting.
Can I say the most spectacular application of Marv's DNA synthesis I've ever heard before? It'll just take a second. So this is a friend of mine, Mike Tyers, at the University of Montreal. And what he's done is wanted to figure out how we could synthesize 1 billion natural synthetic products really rapidly and screen them equally rapidly. So what he's done as a preliminary trial is synthesized 1,600 genes from microbes that regulate natural synthetic product cycles. And what he's done is put them together in cassettes of 4, 5, or 6 in back vectors and placed each of the cassettes in a different yeast cell. So what he has the ability to do then is create, I think the calculation is if you do the combinatorics, 10 million different natural synthetic products. He's demonstrated this works beautifully. And you use the fundamental unit of the cell as selection. So he can put it into high throughput yeast selection techniques, and he can do all of this at a rate that's about 1/10,000th what drug companies do to look at their much more limited repertoire. And let me say, a large fraction, maybe 60 or 70% of the drugs that are out there today are either natural synthetic products or derivatives of them. So it's an incredibly important space to search in, and that's just one example of the power of synthetic biology and its ability to construct global approaches to problems that really have been solved in a very ineffective way in the past.
Yeah.
It's really an advanced entry for the science magic show, isn't it? For the chemistry magic show of the 6th grade.
Well, the cool thing is you can think about making drugs real time to deal with drug-resistant bacteria. I mean, there's so many cool things you can do with that. So.
You know, another example of that is, you know, maybe 2 or 3 years ago now, Ham Smith and their collaborators put together the genome for a very small bacterium, Mycoides, whatever. And they reduced it down to the bare minimum to get this bacterium to function properly. So they took the natural genome, 1 million and some base pairs, and reduced it to around 500,000 by just selecting genes that the microorganism needed to survive. And that was their minimal genome. This is an answer to why would you synthesize a genome? Now, here's the kicker on that. Of those, I think it's about 450 genes, something like that. 1/3 of those genes, we have no idea how they function. So here we've got a very simple bacterium that survives, and we don't even know how 1/3 of those genes function in that little bacterium. And that's the power of working in one direction, and that's making a synthetic genome and seeing what you can do with it.
There's still so much to learn.
Yeah, so much.
I want to make sure we have time for questions. And so if you have a question, you can raise your hand. There will be traveling microphones. So, and if you don't have questions, I have lots more, so don't you worry. But I do want to open it up right now if anyone wants to have the discussion started. And if not, then I'm going to move ahead to another topic. Okay, people, you all lost your chance.
There's one over here.
Oh, boy, that was close, wasn't it?
This idea of how to attack chronic diseases through the systems approach. Can you tell us more about it?
Sure. So the idea is the following. If you take individuals and follow them over time with these dense, dynamic, personal data clouds, what you begin to see after you've followed them for 1 or 2 or 3, in some cases we've followed them 4 years, is people actually transitioning from wellness to a disease. And the most common disease we've actually seen has been cancer. So the interesting point then is because, and right now, the transition to disease is a medical diagnosis, okay? We can change that in the future. But because we have years of blood samples before that, we can go back and examine each of those prior blood samples, and we can look at each of the 1,500 analytes we've analyzed in these data clouds to ask, are there outliers? And what we see with every cancer we've looked at, so suppose we had, in this example, 3,000 individuals, and we looked at one particular protein, we'd see that there would be a very sharp, kind of bell-shaped curve. But what we've seen in every case are outliers. They're way orders of magnitude beyond that normal bell-shaped curve. And in at least 3/4 of the cases, the proteins we identified, one, have a tissue specificity and to a functionality that is actually consistent with part of an oncogene kind of process. So the idea is we can use these outliers either as biomarkers or ultimately possibly as drug targets to see what's going on. And because we've now developed the computational techniques where with every single blood draw, we do all the outlier analyses. And so we're beginning to get all sorts of outliers for which we don't yet have the diagnosis. But we've seen that for diabetes, we've seen it for cardiovascular disease, we've seen it for neurodegenerative diseases. So the idea is then you can get the biomarker that indicates the earliest point at which the transition has occurred. And the really cool idea is for cancer, you can use then immunotherapy in any one of its two modes to erase that very early cancer, maybe before you can even detect it with any methods that you have. So those are the kinds of things we're thinking about. And we're well along doing exactly the same thing for Alzheimer's too. So
there's a question over here. If we can.
Not so much a question, Lee. I think you have to explain the different technologies that you actually look at these people for in their blood samples every year. So we all talk about systems biology, and you talk about these 5 different things you're running, but for the sake of the audience,
you mean
what they are?
Against Data Cloud?
You are, yes.
Okay, so we've done a complete genome sequence for each individual, an enormous amount of data that we're only beginning to decipher. Then we typically take blood draws every 3 months, and we analyze 150 clinical chemistries. We analyze 400 proteins. We analyze about 1,000 metabolites. We do the gut microbiome every 3 months, so we quantify species within that. And then we have digital measures like the Fitbit that is activity, pulse, quality of sleep, how much altitude you've climbed, plus blood pressure and things like that. And what we find is when you integrate that data together in a proper way for an individual, it will lead to a whole list for almost everyone of multiple actionable possibilities, which, if acted upon, will either increase wellness or let you avoid disease. And the really key part of this whole thing is we have wellness coaches that are trained in psychology and the ability to A, understand what an individual's true healthcare objectives are, and to B, put the actionable possibilities in the context of their healthcare objectives. And these healthcare coaches have given us about 70% compliance with actionable possibilities. So they're an absolutely essential part of the process. But the interesting thing is they won't scale because we want to go to billions of people. And if a good coach can handle 100 to maybe 125 individuals. But so I think we're going to have to go to avatars. So we're actually talking with some really interesting groups that are thinking about avatars and how we could make avatar health coaches that could do the same kinds of things that humans can do. And I was just at a meeting with Amazon for 3 days, and there we talked a lot about those kind of things, and Alexa, and the kind of things you can do with that. She has really interesting possibilities for, and they're well aware of it, but they haven't quite figured out which way they're going to move yet.
It's interesting that even in this fascinating vision that you present of automated analysis, that at the end of the day, you're still limited by how many good people you have around you, right? And that's what the
psychology,
right.
For the behavioral change side of it, it's all personal.
It's still very difficult to automate. I know we had a question over here also.
Hello. I have a career advice question. So I'm an undergraduate student, and I know that everybody in the panel is biased, right? You all have PhDs. But I know that there's a lot of opportunity in research, but do you think it's necessary to get a PhD? Is there opportunity within industry to make a difference in biotechnology?
Without a PhD?
Yes, exactly. Sans PhD.
I think you have one answer.
Well, you know, when I was an undergraduate senior, I interviewed some companies. And I had offers. I interviewed 4. I had 4 offers. But during that interview process, I discovered that if I really wanted to make an impact, this was in the chemistry industry. And in those days, I wanted to go into business. Even there, I had to get a PhD. So my idea was I was going to get a PhD and then an MBA, and then I'd go into industry. Along the way, I figured out I liked research better. But I came away from those interviews convinced that if I really wanted to make an impact, I had to become somebody who was in charge of something. And if you don't have a PhD or an MD, and you want to go into research and science, I think you need something more than a bachelor's degree. That's my opinion.
You have that entire side of the room is all nodding their heads that they've all heard you on that. I think you've just increased graduate application rates in some universities. Are there?
I was going to say, great question, Josh. I didn't plant you in the audience, but good job on that.
I'll tell you the real key point is the PhD gives you access to a position where you can control your own fate in a way that it's very difficult to do at a lower level. Now, the exception to that is if you're really good in hacking, you can go into engineering and you can be a non-PhD and you can do quite well. But I think in the more conventional sciences, chemistry and biology, medicine, you want to enable you to be at a position where you can make a difference.
No,
that's the key point.
Just to continue the brainwashing, even if you're a chemical engineer sophomore, you still need to get a PhD. And I'm looking at you. Yeah. Right.
But that's a good choice of undergraduate major. I commend you.
Yeah. Yeah.
And I actually will add to that by saying that I think that in the computational sciences, if you want to be on the cutting edge of writing the most interesting algorithms and doing the most interesting projects, a PhD is still the right way to go. I agree with you that you can get very far as an advanced programmer, but my own huge bias is that if you want to be on the cutting edge, you'd better know your field top to bottom before you go in and think you're going to make it.
Now, the exception to that is if you're infinitely talented, doesn't matter.
That is not a problem that I face.
There are not so many of us out there that are infinitely
Right, that was not my own problem.
Yeah.
But I would imagine that's true. Are there other questions? There's a question in the back, at the table in the back.
I hear that 80% of jobs in biotech are not published online, so you get those jobs by networking. Is that true?
Is it true that 80% of the biotech jobs are done by networking and not posted online? I'm actually not clear why that's restricted to biotech, but it would be interesting to hear your perspectives.
I'll tell you, for academic jobs, you have to list them online.
Of course.
It's a part of the fundamental requirements. I think in industry, it is much more common to do networking and to go to academic centers and say, tell us your 10 best X kinds of things. But for academic jobs, we have to post.
Of course.
Yeah.
I thought in the state of California, you actually had to post a job online. Whether or not that actually means, yes, I thought if you were a company in the state of California, like legally, you had to post in order to hire.
But I do think that there is a lot of value in networking to know which positions are there and where to find the posting once it does appear, which might be after the date you start the networking. You want to know that it's coming. So it is hard to list an industry where networking is not beneficial to you in advancing your own goals and participating in the community as a whole.
And more importantly, just finding out that the type of job even exists. So for example, one of my recent PhD students now works at the FDA, actually doing in diagnostics, evaluating up-and-coming diagnostics. And the particular division she works in at the FDA, I had never even heard of. And I didn't know anyone. But she was at a conference, at an ACS conference, and met somebody who worked at the FDA and networked through them. And then when the job came open, they contacted her. So networking can not only give you a foot in the door at jobs that you know exist, but it can get you a foot in the door at jobs and careers that you didn't even know existed and you weren't even considering, which is perhaps more important.
I will say that when I was a graduate student, we used to say that the extroverts were the ones who were looking at someone else's shoes. And so I think for scientists and engineers, networking doesn't always come super naturally, as in super space naturally. But I think it is probably one of the most important skills you can develop in advancing your career. It's just as important as understanding what are interesting problems to work on and learning how to be stubborn, I think, in working on problems that you think are interesting.
Yeah, I think you have to network. And I think you have to have additionally the right people who will be spokespeople for you. I mean, I get calls, not every day, but on a regular basis from industry asking me if I have anybody or I know somebody who's an expert at whatever. And in other words, there's this system of finding people that doesn't necessarily mean you go to a website and you look at different different job opportunities. So I think there is this subsurface of a networking in its various forms that's really important in terms of no matter what field you're in, not just the biological sciences or physical sciences, but every field it's that way. I'm sorry, but that's the way it is.
Are there other questions? Oh, there's one here. Yes, go ahead.
So could you talk more about pursuing something that isn't as popular, or finding something that you're interested in pursuing and whether or not that intersects with what the world needs? And did you ever find yourself without funding? Did you stick through it? Could you just talk more about that journey?
So who would like to start on the topic of did you ever find yourself without funding? Or I think your stunning no from NIH is such a great story. Yeah. Or other points in your career where you felt that you were working on something that wasn't well received. Is that a fair way to talk to that?
Well, I often did research that was not on my research grant. I had applied for a grant to do something or another. And I ended up doing something else. It's that simple. And as long as you get some interesting results, these places don't really care. As long as you get something that's really exciting, you know? For example, the first group I had working on nucleic acid chemical synthesis, I did that on a different grant, you know? Had nothing to do with that project. And I think that happens all the time. But you know, if you don't produce, you're still in trouble, no matter whether it's on a project that was in your grant or something else. So there's that pressure element in the funding process all the time.
You want to add to that, Andrea, about the problems of creating new opportunities?
So I'm fortunate and not fortunate at the same time. So most of my funding comes from the Department of Defense, where you have to do what you say you're going to do, because I have monthly meetings. It's great. It's like having extra parents. So I don't get that kind of flexibility. On the other hand, and Giannis, my dean, might get mad at me in about 2 seconds, I have the additional flexibility of I can teach an extra class and then convert those teaching units into money, which I don't know if I'm supposed to advertise that. Okay, yeah, it's too late, cat's out of the bag. And then I can come up with money that way. So I can teach more and get money and then use that money to fund pilot projects. So I can literally work harder and get money. And it's a very straightforward way to work harder and get money. And at least it's a sure bet.
Based on what you just said, I'll cross Department of Defense off my place to apply.
Every Tuesday, first Tuesday of every month at 6 a.m., I have a conference call. Yeah, great.
Definitely, I'll mark it.
Yeah. But so that having at least a wonderful dean who's right here really at least enables me to pursue kind of the crazy ideas. And without that, without having at least some form of support to do the truly outrageous ideas, to at least get the prototype data so that you can then get the larger funding from an NIH or an NSF, though that hasn't happened, or the DoD that you get micromanaged, that has happened. That's critical.
I think one of the most important attributes of a scientist is determined optimism, because you're almost always going to run into criticism of one sort or another. And look, if you're right or you're convinced you're right, I think you just have to power through those kind of things. And it's character building. Mm-hmm. And let me say that there are a lot of sources of funds too, and you don't have to just say the dean is the only source. Well, maybe I'm getting in trouble with the deans too, but at Caltech, I talked to people who gave philanthropic money. I talked to foundations. I certainly got support from industry in addition to federal grants and contracts. So there are alternatives if you've got a great idea and you're not getting either government funding or the local funding you need to do it. Don't give up. Go find somebody and get them excited.
But you're really highlighting the personality element, which is that it is very hard to be disruptive or develop disruptive technologies without disrupting something. And so you need to have a certain element in your character, which is, I am going to figure out how to make this happen. That's what the determined optimism is. Yeah. Did you have a question? I think a microphone will come to you. It's a big room, though.
So I would like to go back on the challenges of teaching system thinking and system biology. So I'm Italian, I'm European, and I see a difference in the way of education of the young student. So I have 16 years old, student coming in the lab, and they already have their path done. So I think they make also their opportunity to learn other things a little bit more smaller, because they already focus on something. Do you have any suggestion how to build your career in terms of education, and if it's worth it to explore different opportunity in different fields?
So your question is how to explore other fields if you want, if you want to broaden your horizons and you want to be more interdisciplinary. Is that a fair way to say it?
Understand system, we were talking before, understand system thinking.
So that you can teach your students. So is the question, how do you manage your own career, or how to
teach the students seems very compartmentalized?
When the students are so compartmentalized in their education program, and I think we talked about it a little bit earlier in terms of how to support systems thinking, because that was such a universal theme. But Andrea, you talked about the different people interacting with one another and teaching engineers that cells die, which I think is a really great lesson. Do you also encourage them to go take other classes outside their disciplines? Do they find additional mentors? How else do you do it?
So if you're thinking about a 16-year-old, right, they have a very structured curriculum. And undergraduates have a similarly very structured curriculum. So then they can get kind of that breadth in what they do in their extracurriculars, right? So if you have a high school student who is, taking all of their classes in, whatever, AP Calculus, AP Biology, AP Physics, then maybe their extracurriculars should be something like the newspaper, right? So something, the opposite of that, right? And when I was in high school, I was on the newspaper, right? It was actually, looking back, it was a brilliant decision because I'm a better writer as a result. And being a good writer is a key skill in being a faculty member. So trying to force the students who think that they don't want to do something because it's not worth their time and they're super busy can actually be one of their strengths, you know, a decade later. We're going to pretend I'm 29. Good, all of you are laughing. This is good. So that would be like if you're a high school teacher or you have a 16-year-old. No, okay, good. You train 16-year-olds, yeah. So if you're training a 16-year-old, that's what I would encourage them to do, is to do something that goes kind of against their grain, whether it's like play a sport, take a studio art class, something just completely different that will really push their personal envelope. Just like, you know, my husband and I do hobbies to try to do something different, and we took a pottery class. Not a skill. But it was fun. But no one will ever see the pottery. But it's fine.
A challenge. Will I get to see the pottery? I like that. Probably not. Are there any other questions? There's a question in here in the third row.
Hi. My question's about something that may be a little foreign to all four of you, but the idea of failure, whether it be in
Sorry.
whether it be on a collegiate level or in industry and in research, what have you found that you've learned that's most valuable about failure?
About failure.
About failure. Well, my list is very long. In case you all don't have any, I have plenty to share, but I have a feeling that everyone has something to say on it. Look, everyone's reaching forward.
I can just say that in research, maximally 20% of your ideas work, more like 10%. And you've got to be prepared to expect something doesn't work. And if you're not prepared for that, you should not be in research. It's just that simple.
I would say that the most important thing about failure is to try to look for the lesson that you can learn from the failure, because you can look at a failure as like a wasted opportunity, right? But in every failure, there is some lesson you can glean from it. There's something you can learn, even if it is truly a failed test, right? That test, that F on a test, is telling you the information you, in fact, did not know. So every failure is actually an opportunity to figure out what you could do differently next time. So it shouldn't necessarily be viewed as a negative. It should be viewed as a stepping stone towards your success. You just need to figure out what that next stone is.
Another comment that I often make to students in the lab, you want to set up an experiment so you get a yes or a no answer. Either one is extremely valuable. But if you get a maybe, that just means you didn't put the right controls in the experiment. And so you've just lost ground. But a yes or a no, and I don't care which way it goes, but that gives you a clue as to what to do next.
And to add to what to do next, I think an important lesson from the lab is don't just do something, stand there, right? Sometimes don't go run another experiment, right? Sit and actually think about what it is that you did and actually organize your data. I think one thing that I see commonly in students of all levels, and you see it sometimes in industry, is that people think it's better to just do more and more tests without actually thinking about what they learned from the tests they already did. And they don't take a minute and gather themselves, gather their chi, and think again about what to do. It actually turns out to be the faster path, I think, to be more thoughtful as you go.
There's actually a great, I don't know if user manual is the best description of it, on intelligent experimental design that's on NIST's website. It's about 200 pages long, but it is meticulously written for any students in the audience. And it's on how to design an experiment, how to check, make sure you have controls in the experiment. And it was probably written based on the website in like 1985. But good experimental design hasn't changed since 1985.
Earlier than that.
Yeah,
that's right.
exactly.
Probably hasn't changed since the Greeks. We just use different instruments today.
Exactly.
I think we're running out of time, and so I want to give each of you a moment to share some final lessons with us. And maybe we'll start at this end. Marv, if you want to share, especially some of the comments you made were so interesting and really touched on leadership skills that are just as important as technical skills. maybe if you have some final thoughts on how a scientist can develop leadership skills also.
Well, to begin with, you're not trained to be a leader in science. You have to develop that skills. And that's really important because when you're working, I'm talking about just strictly science. Mm-hmm. When you're working in science, you have to be a leader, irrespective of whether you're a group leader, you have your own research group in a laboratory, in a university, or in a company, or whatever, or if you're a department chair, or you're whatever. You don't take classes in how to be a leader or how to manage the classroom or your research group or your department, but you have to develop those skills, and if you don't, the chances are pretty good you're never going to be someone whose students are going to say, yeah, I want to work with that person. Boy, that person really knows what they're doing. And there's as much leadership there as there is good science, because you can attract students to your lab if you can communicate with them. And that's not easy, because some students, well, everybody's different. And so you have to communicate in a different way to each and every student, or if you're a department chair, with each and every faculty member, et cetera, et cetera. And there aren't any courses in that. You have to learn how to do that as you grow from the classroom as an undergraduate or a postdoc or a graduate student and take on a role in some profession. You have to learn how to do that. You can't take a course in how to become a leader, shall we say. I'll stop at that point.
Although it's an interesting point to stop, given that if you can't take a class and you have to learn, that's a pretty daunting task.
But you have to do it that way, and if you can't, you're not going to be a very good leader.
That's right. So, Andrea, the room is full. I presume these are all your students.
They are, yeah. I have a really
This is about
large...
the size of your lab, is my understanding.
Yeah.
So if you have some additional thoughts on basically how do you go from the student stage to really having a compelling group that is doing so many different things. You know, you started by talking about your passions and your passion for teaching. How do you maintain that level of passion for your science every day when you walk in, and the same level of passion for your teaching every day?
I mean, honestly, I kind of view myself almost like a scientific vampire. I draw my passion and my energy from my students, right? They come into work every day and they're excited to be there, they're enthusiastic to be there, and then I get excited and enthusiastic to be there. And then it's like a feedback mechanism, right? We just get more and more excited, and it's kind of dangerous in that sense because there isn't really any kind of control loop, any stop function on that. The stop function actually is the rejections, right? Like when you get a rejection from NIH or a rejection from a journal, right? Like that's like the stop function. Right. Otherwise, it might just spiral out of control, and that's when we get free food on Fridays. But it's that just kind of environment of just excitement of discovery, and that's what keeps me coming back to work every day. Just being in an ecosystem where every day something great is going to happen, and every day something new will be discovered. And maybe it'll be a baby step, but maybe it'll be a gigantic leap, and you never know. And so that's what makes this a great place.
And the great thing about your statement at the beginning, it wasn't clear if you said scientific vampire or scientific empire. And I thought both really apply equally well to how you come into work in the morning. So I think it's terrific, though, to draw so much energy from the people around you. I mean, it clearly works as a feedback loop. Did you want to add something?
I was just going to say, you know, one of the rewards I really enjoy is, you start with a new graduate student, and they're so tentative usually, and they really don't understand what in the world they're doing. And they come in with the result, and you say, oh, gee, that's really interesting. Why don't you repeat it, and let's see what happens. And usually the next day or the next week, it's, oh, you know, that didn't work, and I can't figure it out. And you kind of go around and around on this theme for quite a while, and then pretty soon they figure out how to get good results. And then, you know, by the time they've been in the lab for 4 years, it's like, yeah, I know what I'm doing. And you can't stop them, and they're on their way, and that's the way it goes. And that's so much of a reward to see that happening to a student that I have to admit that's one of the most favorite parts of being a research director.
Yeah, I get energy from them. It's.
And I thought of that relative to what you just said.
Yeah.
And Lee, if you want to give us some additional thoughts on the process of just bringing an idea, a large vision, and thinking it through and being able to communicate it so crisply. You talked about the four Ps, or the P4 approach, and how that helps guide what you do every day. You know, how do you really come to terms with that vision when it starts as just a series of ideas? How do you bring that together?
Well, so let me take a slightly different tact, but let me give you my algorithm for science and why I've enjoyed it. I think, one, you have to do things that give you enormous pleasure. And having a good time and being excited about what you're doing is really key. I think for me, what you mentioned there, I think it's really important for people who want to make contributions to think about big ideas, to think about important ideas, to think about something that really is going to make a difference. I think it's really key to work with the best possible people you can because they set your standards very, very high. I think this idea that very few of us learn, which is really critical, and it's almost cliche today, thinking outside of the box is really critical if you're to get into big new kinds of ideas and so forth. And I'd end up saying, if you flavor all of this with determined optimism, you'll really have a terrific career.
I think that is a note that will have everyone here leaving with determined optimism.
Good.
So thank you all for your attention this evening, and thank you especially to our panel.
Yeah.
Well, I was extremely impressed. I'm sure all of you were as well. Thank you all for coming. So this event is one part of an Evening With event series. We host all of them at academic institutions across the country. We've had 2 this year. We have 6 more coming up. Please come to our website, nationalmedals.org. Find out where we're going to be next, what laureate or laureates we're going to feature. If you can't make it in person, we do a live feed, so feel free to tune in when those are up. A couple people to thank here. First and foremost, I wanted to thank Amgen for sponsoring the event. The contribution was incredibly valuable to us. I'd like to thank Dean Yortsos, the USC staff, Dean Bradford. Thank you very much for putting this together. I'd like to thank the United States Patent and Trademark Office. They provide us with valuable support as well. Dr. Belz, thank you so much for doing such a great job moderating. You were fantastic. And finally, let's give a round of applause to our panel members again, Dr. Caruthers, Dr. Hood, Dr. Armani, thank you very much. There is still food left. Please go eat. Continue your conversations. This is a really valuable time to continue that dialogue. I think there's a few more things of swag left. I don't really want to figure out how to check a bag on the way home, so please take everything we've got on that table. You know, we don't baggage claim at DC Reagan is really tough anyway. So have a good day. Thanks a lot.